Thank you all for being here this afternoon. I've really been enjoying my time here as a visitor at Nimbus and can't believe that it's already almost November and there's so little time left. But anyway, I hope to meet more of you as the remainder of the semester goes on. So the work that I'm talking about today actually started while I was a graduate student at the University of Florida. These individuals and myself, we were all part of an IGERT fellowship program. So that's an NSF-funded program. It stands for Integrative Graduate Education Research Traineeship. And so the work started with this group of individuals: Miguel Acevedo, Trevor Coughlin, who's now at Boise State University, Kenny Lopiano is the founder of Roundtable Analytics. He's a statistician, and Nick Rukta-Nunchai is now at Southampton. But I'm continuing to work with Miguel and Nick on this work. So Miguel is now an assistant professor at the University of Florida in wildlife ecology, and Nick is at Southampton in geography, and he's doing work on spatial epidemiology. So I'll start with a bit of background about malaria and one of the first models for malaria, or mosquito-borne disease in general. And then I'll extend this model to include spatial heterogeneity. And those regions in space that have different characteristics are going to be connected through human movement, either migration or short-term movement. And in the short-term movement model, I'm also going to look a little bit at individual heterogeneity. And then I'm going to talk a little bit about how we can move from theory to practice using some cell phone data from the Dominican Republic and some community detection algorithms. So malaria is a vector-borne disease transmitted by female Anopheles mosquitoes, and the disease in humans is caused primarily by one of 4 species of the Plasmodium parasite: Plasmodium falciparum, Vivax ovale malariae, and then there are some known cases of no lessee in humans. So this requires both the vertebrate host, such as a human, and mosquito to complete a full life cycle. The World Health Organization estimated that there are about 216 million new infections in 2016, resulting in almost 500,000 deaths. And most of these deaths occur in children under the age of 5, and the major cause of that is Plasmodium falciparum. There's actually been a big reduction in the number of cases over the last couple of decades due to pretty intense efforts in terms of malaria control. But there has been a slight uptick in the last couple of years. And these numbers, of course, are still very high, so there's still a lot of interest in trying to understand what kinds of factors might be affecting the efficacy of these various control strategies that we are currently using. So the discovery that it was mosquitoes that transmit malaria to humans was made by Sir Ronald Ross in the late 19th century, and then he published the first mathematical model for malaria in 1908. And this was later extended by George Macdonald in the 1950s. So in the literature, there are a lot of slight variations of these mathematical models, and so we often refer to this set of models as a Ross-Macdonald framework. So one example of these models illustrating below here. So the basic idea is to divide up the human population into 2 groups: susceptible humans and infected humans. And similarly, we can divide up the mosquito population into susceptible and infected vectors. The only way that a human can become infected is if they're bitten by an infected mosquito. So that's denoted by this dashed arrow here. And then we have to figure out how to express the rate at which humans become infected. So one quantity I want to draw your attention to is this product M times A. So M is the ratio of mosquitoes to humans, which I'll come back to later on, and A is the biting rate. So this product is the human biting rate. And so the nice thing about this quantity, it's the number of bites an individual receives per unit time. And so this is something that can actually be measured in the field. So you can stick somebody outside and wait and see how many times a mosquito bites them, and you get this nice parameter, M times A. But not all bites by mosquitoes are going to be by infected mosquitoes, so we need to multiply by the probability that that contact is with an infected mosquito. Here we're assuming that the mosquito population is well mixed, so the probability that that bite is by an infected mosquito is simply the number of infected mosquitoes divided by the total mosquito population, so the proportion of the mosquitoes that are infected. And then with some probability B, then transmission actually occurs. So if we multiply this by the total susceptible humans, then this gives us the number of new human infections per unit time. The model also assumes that there's no lasting immunity to reinfection. So the individuals who are infected, once they recover, they return to that susceptible class at some rate. So we can come up with equations for the mosquito population in a similar way. Again, we can write down an expression for the rate at which new mosquito infections arise. And again, that can only come about if an infected human takes a blood meal from this infected mosquito. Those susceptible mosquitoes progress to this infected state. And then because mosquitoes have such a short lifespan, it's often assumed that the mosquito dies due to natural causes rather than as a consequence of the disease. And so the mosquitoes die at this natural mortality rate given by G. So we can take this picture and write down a system of differential equations where each equation here represents the rate of change in one of these variables. So for example, the second equation represents the rate of change in the number of infected humans. We can make a couple of observations to reduce the number of equations here down to 2 equations. So one is that because we're ignoring disease-induced mortality in the human population is actually constant. And so we can omit the susceptible equation and rewrite the susceptibles in terms of the total human population minus the infected. And we can also express the mosquito population as a proportion. So Z now represents the proportion of mosquitoes that are infected. So now we have this system of 2 equations that demonstrate that represent the dynamics of this system. We can also group together some of our parameters. So now I can define the transmission rate alpha from human to mosquito by alpha, and beta represents the transmission from mosquito to human. So again, this quantity M is going to become important later on. M is, again, the ratio of humans to mosquitoes to humans. So total mosquito population divided by the total human population. So one of the things that we can derive from this simple model is the basic reproduction number. So this is a concept that most of you in the audience are probably already familiar with, but it's the number of secondary cases resulting from a single infected individual in an otherwise fully susceptible population during their time infected. And so the idea is that if we take one infected person, introduce them into the population, if the reproduction number R naught is equal to 2, let's say, then that person will on average infect 2 more people. Those 2 individuals will go on to infect on average 2 more, and so on. So we get this idea that the infection should spread if the reproduction number is bigger than 1, and that it should die out if it's less than 1. And mathematically, it's a threshold quantity associated with the stability of the disease-free equilibrium. So if it's less than 1, the disease-free equilibrium is locally asymptotically stable. So in most models, this will mean, or most simple models, this will mean that the disease will die out. And then if R naught is bigger than 1, the disease-free is unstable, and the disease can spread in the population. So this is quite easy to derive for the Ross-Macdonald model. We can write it as a product of 2 terms. So the first is the number of infected humans per infected mosquito, and the second is the number of infected mosquitoes per infected human. So this reproduction number matches our epidemiological definition. It's the number of new infected humans per infected human. And so the nice thing is that we can study something like this to understand what kinds of measures we can take to try and push the system towards elimination of the disease. And so in particular, one of the things that was discovered by studying this model and this quantity is that it's not necessary to get rid of every mosquito in the population in order to eliminate a mosquito-borne disease, but instead we need to do things to reduce this reproduction number below 1. And so we can do that by either increasing the mosquito death rate, increasing the human recovery rate, or by decreasing the biting rate using something like bed nets. And we can also easily calculate the prevalence of malaria in humans at equilibrium. So this is just the number of infected humans at equilibrium, or the proportion, I should say, of humans infected at equilibrium. So if you plot this as a function of R naught, you see that this is always an increasing function. And so this is going to be important for us later on. So this model has been useful in terms of guiding some decisions about malaria control, but as I mentioned, the burden is still very high, and so we need to better understand other factors that might play into the efficacy of a given control measure. And so what motivated our work on studying human movement in these mosquito-borne diseases was looking at pictures like this. So this is Hispaniola. On the west side, we have Haiti, and on the east side, we have the Dominican Republic. And the darker red represents higher malaria prevalence. And so we can see that the prevalence of malaria is very heterogeneous across this island, and there's a pretty stark contrast between Haiti and the Dominican Republic, with transmission being pretty low in the Dominican Republic. The black lines represent roads, and then these yellow plus signs represent border crossings. So this picture might look different now because this is from a while ago, but the idea, though, that we want to convey here is that we see higher prevalence in the Dominican Republic along these regions where we have roads and near these border crossings. So this gives some indication that human movement is playing a role in the distribution of malaria prevalence. So there's different factors going on. Part of it are the local transmission, the local conditions that might promote transmission more in certain regions of Haiti, but also human movement is likely also playing an important role. And so the idea was to initially abstract this and consider the simplest case where we have two regions with different levels of transmission. So we can consider patch 1 as being a high transmission region and patch 2 as being a low transmission region, for example. And here we have essentially Ross-Macdonald type dynamics going on within each patch. Here I'm just showing the human population. And then we have migration of humans between these two regions that connect the two regions. So here, a migration rate K21, for example, means the rate of movement from patch 1 to patch 2. So the second subscript is where you're coming from, and the first subscript is where you're moving to. If these two patches are isolated from one another, we can derive a basic reproduction number that looks exactly like our reproduction number for the Ross-Macdonald model. So we're going to assume this one is larger than the one for patch 2. But as soon as we introduce migration, then now we have a reproduction number for the entire system that I'll refer to as the global or network reproduction number. And so this is what the quantity that's actually going to determine whether that disease-free equilibrium is stable or unstable. So you can derive this using that next generation approach, and so you get this more complicated expression. But we can still use this to try and understand how the reproduction number depends on quantities like the migration rate. So here we've plotted the global reproduction number. So this surface is the global reproduction number as a function of our two migration rates. And it's sandwiched in between the two isolated patch reproduction numbers represented by these two planes. So the isolated patch reproduction number is below 1 in this example, and the reproduction number for the high transmission patch is above 1. And so you can see from this surface that there's going to be some values, some rates of movement between these two regions that allow the disease to spread in both populations. So essentially by connecting two regions together, that can allow the disease to persist in a region where it would otherwise die out. So the next step was to extend this to more patches. So in the previous example, you could think of one patch as being Haiti and the other Dominican Republic, or it could be two villages with different degrees of transmission. But you can, of course, think of examples where you would want to extend that to more patches. So again, we have the rate of change in the proportion of infected mosquitoes, the rate of change in the, and this is in patch I, rate of change in the number of infected humans in patch I, and then the rate of change in the total human population. So we can introduce the migration, so this would be immigration here from all of the other patches into patch I, and then here we have emigration from patch I to all of the other patches. And we made some assumptions about movement. So here we're assuming that movement is constant and symmetric across the landscape. So that means replacing all of these migration terms, the kij's, with the same parameter k. So for any two patches, the rate of movement is the same in either direction. So by doing this, it makes the total population size constant, and so we can again reduce the system significantly. But we needed a metric for how to actually quantify heterogeneity. So we used this coefficient of variation, a common metric from landscape ecology, to do this. And we defined it here as the ratio of the standard deviation in the ratio of mosquitoes to humans across the landscape, and the mean of the ratio of mosquitoes to humans across the landscape. And then we needed to construct our networks in some way. So here we chose 10 patches for each of our networks, and in each of these figures represents a scenario in which we have increasing heterogeneity in our network. So we start out with the coefficient of variation equal to 0, so that's the completely homogeneous case, and then we systematically increase the amount of heterogeneity. So for each of these coefficient of variations, we actually have 100 different networks that we constructed. And we did that in the following way. So we make the transmission different in each of the patches by choosing different ratio of mosquitoes to humans for each of the patches. So for a given network, let's say that I'm looking at the case where we have the highest degree of heterogeneity. We construct this network by drawing the ratio of mosquitoes to humans for each of these patches from a normal distribution with mean 60, and in this case, standard deviation of 40, our highest degree of variation. And by doing it in this way, it means that if we were to take the average of the reproduction numbers for each of these patches, we get approximately the same value for each of these networks. Okay? So essentially, altogether, we have 400 heterogeneous networks that we simulate with one of these 4 different levels of heterogeneity, and then we also, of course, have the homogeneous case. So I should mention, by doing this, by choosing the ratio of mosquitoes to humans in this way so that we keep the average of the reproduction numbers, the patch reproduction numbers, the same across all of the networks, that allows us to tease apart the effects of things like the rate of movement and heterogeneity. So we can first look at the global reproduction number, or actually maybe I'll start over here on the right side. So we've got our isolated patch reproduction number for our different increasing degrees of heterogeneity. And so you can see here that the average across those isolated patch reproduction numbers is approximately the same. Okay, so moving back to this first figure where we're looking at the global reproduction number as a function of the rate of movement. So as this rate of movement k increases, the strength of our network is becoming more connected. So we find that the black line here is the homogeneous case. So of course, in the homogeneous case, the movement doesn't matter. And the reproduction number in the other cases is a decreasing function of the rate of movement. If we look at what happens as we increase the coefficient of variation, then we see that the reproduction number actually increases. And so we see the same thing when we look at prevalence. So here, prevalence I'm defining as the proportion of individuals infected at equilibrium. So again, as you increase heterogeneity, you increase prevalence. But what we see here is that this is not happening in a monotonic fashion when we look at it as a function of movement. So as the network is becoming more connected, it turns out there's actually some intermediate rate of movement or intermediate level of connectivity that maximizes disease prevalence. So what we think is happening is that when people start moving around, now some individuals are being exposed more to the high transmission patches, so this can increase the number of people who get infected in the entire network. But then after a certain point, you're moving so quickly that you have less time to get infected in those high transmission patches, and so things start to decrease and then level off as things get more and more mixed. And just to give you an idea, this rate of movement of 0.1 would mean that people are moving every 10 days. So this is actually quite fast if we're talking about migration. So the more realistic values of migration are going to be on this lower end. If we look at prevalence as a function of the reproduction number, we get a very different picture from what we saw in the homogeneous case earlier. So remember I showed that bifurcation diagram where you have the prevalence was always an increasing function of R naught and then starts to level off. But now when we include this spatial heterogeneity in transmission and human movement, now we get these hump-shaped relationships between prevalence and R naught. So again, it's some intermediate value of R naught that seems to be maximizing prevalence. And so this is highlighting the fact that if we were to make decisions based on the reproduction number alone, that's really not enough information to be able to say something about disease prevalence. We need to know something about the degree of heterogeneity in the region and how that region is connected. So then we got to thinking about whether similar results would hold if we considered different types of movement. So what if we consider short-term movement instead of this long-term migration? And are there other types of heterogeneities that might produce similar patterns? So we already saw some evidence from, so one of my collaborators was looking at individual-based models that had short-term movement, and there was already evidence in there that some similar patterns were emerging as far as this non-monotonic relationship between metrics for prevalence and the reproduction number. So we wanted to explore this a little bit further in a systematic way. So we are looking at, and this is ongoing work right now with Nick Ruchtanunchai and Miguel Acevedo, and this is what we were working on over the summer here at Nimbus. So we're looking at these different cases. So of course we first have this homogeneous case. So we have homogeneity in spatial transmission and individual risk. Then we can look at the case where space is homogeneous but individual risk is heterogeneous. Then we can look at the case where we've got spatial heterogeneity. So this is essentially what we have been looking at, but now we're looking at short-term visits to other patches rather than permanent migration. So we assume that an individual in one patch spends some proportion of their time in the other patches. We can look at heterogeneity in movement. So the movement may not be symmetric anymore. We can then combine different types of heterogeneity. So we can combine heterogeneity in individual risk and spatial transmission, and we can combine heterogeneity in movement and spatial transmission. And of course, in principle, we could combine all three, but we focused on these 5 cases and wanted to compare these 5 cases to the homogeneous case. Yeah, so for the individual, we group individuals into different risk groups. So it's not truly individual in the sense of some kind of agent-based, right, more groups, right. And so that heterogeneity could be influenced by a number of different factors. One would be age. So different age groups have different immune systems, and it could also be generated by things like what kind of work the individuals are doing. So certain types of jobs make you put you more at risk for exposure. It could be where you're living, if you're closer to breeding habitats or things like cattle that might attract mosquitoes. So there are a number of different things that might influence things like individual risk. So we can set up our system of equations in a similar way. So now we're keeping track of the number of individuals of a particular risk group in a particular patch. And now instead of tacking on those migration terms at the end, because we're modeling short-term visits to other regions, we now incorporate that type of movement into the incidence term. So the incidence is the product of the force of infection, so that's given by these lambda terms over here, and the number of susceptible humans, or in the case of mosquito infection, we multiply by the number of infected mosquitoes, or susceptible mosquitoes. So let me move forward to, so we make some simplifying assumptions first of all. So here, the P's represent the proportion of time that is spent in other patches. The Q's represent individual infection risk. Sigma is the biting rate, and betas are the probability of transmission. This exponential term is related to the probability that a mosquito survives the incubation period of the parasite. So we assume that the way that we're creating the heterogeneity in this case, instead of changing the ratio of mosquitoes to humans, now we're including explicit demography. So we are now creating spatial heterogeneity in transmission by allowing the birth rates of the mosquitoes to be different in different patches. So we keep all of the other parameters associated with each of the patches the same. And so by doing that, this simplifies some things, so we can pull out some of these coefficients, and we can write all of this as some products of matrices, where P here is the matrix of the proportion of time that individuals spend in each of the patches. So along the diagonal, you have the proportion of time you spend in your home patch, and then the off diagonals are the proportion of time you spend in the other patches. And then Q is a matrix with the diagonal entries equal to your infection risk. And so each of those entries is going to be some quantity. Actually, in each case, the entries are going to be between 0 and 1. Okay, so we want to compare those 5 scenarios that I mentioned to the homogeneous case. And so the way that we do that is by pairing up our simulations according to their reproduction numbers. So for a given point, we have a particular reproduction number and the corresponding prevalence for the heterogeneous scenario and prevalence for the homogeneous case. So if all of the points lie on the y equals x line, that means there's no difference in prevalence between the homogeneous case and the heterogeneous case. If the points lie above this y equals x line, then that means that prevalence is higher in the heterogeneous scenario than in the homogeneous scenario, and then we have the opposite conclusion. Heterogeneity is lower in the homogeneous or in the heterogeneous scenario. So if we think back to the scenario when we were looking at migration versus instead of short-term movement, that would put us in this middle situation where heterogeneity increased prevalence. So now we want to see what goes on when we look at short-term movement and the impact of heterogeneity in risk and spatial transmission. So we have our 4 different scenarios here. So we've got the heterogeneity in space, heterogeneity in movement, and I should mention here these patches should actually be different colors and the people should be all the same color. So this is actually supposed to be the case where you've got heterogeneity in spatial transmission, but all of the individuals have the same risk. Then we have heterogeneity in space and risk, and then finally we have heterogeneity in just risk. So in all of the cases, we see that these points are falling below the y equals x line, which is contrary to what we saw before. So now this heterogeneity is actually decreasing prevalence. So the homogeneous case is overestimating prevalence compared with the heterogeneous case. So in other words, it's kind of represents the worst case scenario. And the more variability that we have, the further away we get from that homogeneous case. This also answers a bit our question about whether we're going to see those similar non-monotonic relationships between prevalence and R naught. So here we're plotting prevalence against prevalence, but here prevalence on the horizontal axis is increasing with R naught. So we can infer from this that the prevalence of the heterogeneous case is non-monotonic with respect to their global reproduction number. So we are working on simulations right now to try and better understand what's producing these non-monotonic relationships. And so far, it seems that individual risk is really what's driving these different patterns, these non-monotonic patterns. We also calculated the error between the simulated prevalence in the heterogeneous case and in the homogeneous case. And so what I want to point out here is that it might not be surprising that the biggest error is in the case where we have both spatial and individual heterogeneity. But what I want to emphasize is that if you were to add up the bars from the individual case and the case where you have spatial heterogeneity, that sum is less than what we get here. So this is something worth exploring a little bit more. It's an indication that maybe these 2 types of heterogeneities are interacting in some way, that it's actually amplifying their effect on prevalence.
What does variability mean
The variability is in, for example, if you're talking about individual heterogeneity, then how different the infection risk is amongst those individuals. So what I've talked about so far, oh, yeah, sure. Well, error in the sense of how different it is from the, yeah. Right, so we use the calculation we use is the root mean squared error, but really it's, yeah, how different is it from the homogeneous case? Yes, thank you. Yeah. So what I've talked about so far has been pretty theoretical. The models are fairly simple and in their structure. One of the things that we were interested in is trying to understand how would you actually come up with these patches? How do you determine what the appropriate regions are? What people do you group together? And how would you actually estimate the rates of movement between these different patches? So one thing that we started looking at was this cell phone call data set in the Dominican Republic. So this data set included 177 towers. There were 57 million calls during this time period, which represent the edges in our network. The towers are the nodes. We have information about the location of each of these towers. We know the number of calls or the call volume between any two towers. We omitted the repeater towers from the analysis because these are towers that are boosting the signal, and we wanted to know where the calls were originating from and where they were going, where they were received. There are, of course, some limitations to a data set like this. So in our case, it only represented a 5% market share for the Dominican Republic. And so it also brings up the question of whether, cell phones are uniformly distributed across individuals, and so of course it's not completely uniform, although it turns out that it does service a wide range of urbanization, wealth, and land use. We also wanted to look at some place-based attributes. So we looked at sugarcane, urbanization, wealth, and distance to airport. And our goal was to see whether we could use the cell phone data and these place-based attributes to determine community membership. So which of these towers belong to the same community. The idea being that people who talk a lot with each other are perhaps more likely to interact with each other. So another of the shortcomings is that cell phone towers, it turns out, are not uniformly distributed across space, and for good reason. And so we wanted to know, when we do our analysis, we wanted to know, well, how much of our community structure is coming from the fact that some of these towers are close to each other. So first, we used a k-means clustering algorithm to categorize the towers into communities using just the distance between the towers. And so as you would expect, the towers that are close to each other are grouped together into the same community. And one community I want to draw your attention to is this one, which is around the capital, Santo Domingo. So there, all of the towers are clustered into the same community because they're all close to each other geographically. But now we want to use some other way. We want to use the call data itself to see if there's any additional structure in the data beyond just the geographic structure. So we used a method from social network theory called modularity. So the idea here is we're going to denote communities by C. So this is just a group of nodes or towers in the network. And P denotes a partition of the network or some arrangement of those towers into communities or modules. And then for each of these these partitions, we can calculate something called a modularity score. So the idea is that this modularity score is calculated as the difference between the observed density of edge weights within communities and the expected density of edge weights within communities. So the idea is that you are comparing the actual call data to some kind of null model where the edge weights are the same, but you're distributing your edges at random. But the challenge here is that there are a lot of possible partitions, especially if you have a large number of nodes. So there's too many partitions to actually calculate the global maximum of this modularity score. And on top of that, there's more than one partition that might give you the same modularity score. Okay. So one of the things that we use this simulated annealing algorithm, and to try and get around this second point that there might be multiple partitions that give you essentially the same modularity score, we repeated this modularity optimization 100 times. So then the output then of each of these 100 simulations is some kind of partition, so it gives us community membership of each of the towers in our data set, and it gives us some modularity score for that particular partition. And so this is going to be a number between 0 and 1, where 0 means that it's not significantly different from the null model. So from those 100 modularity optimizations, this picture here gives us the partition with the highest modularity score. And so now, if we look back again at those towers around Santo Domingo, they're no longer clustered into the same community. Now there's this clear division between the west and the eastern half of this city. And our collaborator, Trevor Coughlin, actually visited during this time and noted that there is a stark difference in socioeconomics between these two sides. So although they're geographically close to each other, they're not talking with each other as much as they are within their communities. And then we can see some other examples like this. If you can see these faint pink dots, they pop up in places that are geographically distant from one another, but they are certainly communicating a lot with one another. So it gives us an idea that, well, maybe these individuals are more likely to visit people over here, even though they're geographically distant from one another. So we use this method to find community structure in the call volume data. We want to see whether these other variables, these place-based attributes, can predict the same communities. So each of these disks represents a 15-kilometer radius around each of the towers. And so if we look at sugarcane, for example, we calculated the area of each of these disks that is covered by sugarcane. And we did something similar for urbanization. So our predictor variables are sugarcane area, urbanized area. For the wealth index, we looked at which municipio each of the towers was in, and then assigned the wealth index of that municipio to that tower. And we looked at distance to nearest airport. So the idea here is to use linear discriminant analysis to figure out what combination of these variables best separates the classes, or best separates the towers into communities. And by communities, I mean those determined by the modularity optimization. So essentially, we repeated this again 100 times for each one of those 100 partitions that we got as an output of the modularity optimization. So on the horizontal axis here, we have modularity. On the vertical axis, we have the predictive power of the linear discriminant analysis. And so what I mean here is, so for example, if you have this modularity score of 0.5, then the predictive power was in some cases was close to 70%, which means that the LDA predicted that correctly placed 70% of the towers into the appropriate communities. And so we were happy to see this linear relationship because it gave us an indication that the discriminant analysis was doing what we wanted it to do. So a lower modularity score presumably means that the partition that came out of this is not as close to the actual community structure as a partition that has a higher modularity score. So if the LDA is doing the right thing, or if these variables actually can predict community structure, then we would expect it to do a poor job when the modularity score is low, and a better job when the modularity score is high. And we also found that sugarcane and urbanization did the best job of predicting the community membership. So to summarize, so first going back to the results from the more theoretical models, we demonstrated that assuming spatial homogeneity can underestimate prevalence in the case where you have migration connecting these different regions. It can overestimate prevalence when you have short-term movement, and it can overestimate prevalence if there is individual heterogeneity in risk. And then beyond that, we saw that heterogeneity leads to complex non-monotonic relationships between, not just between prevalence and R naught, but also between prevalence and movement. And so this means that when we're trying to make, if we're trying to make predictions or trying to determine the risk of a particular region, we need to take into consideration these different factors. On the side of the community structure analysis, we found that both the mobile phone data and these place-based attributes could be used to identify these communities. We saw that there was more information in that call volume data than what you get just by looking at the spatial location of the towers. And then finally, because we could use those place-based attributes to predict community membership, at least to some extent, it means that this might give us some way of trying to extrapolate community structure in regions where we don't have these cell phone towers. So there's some more people that I need to acknowledge. So in the beginning of our work, when we were all still graduate students, we had a number of different mentors that helped a lot in all of this. So Nathan Eagle provided us with the cell phone data set. Andy Tatum is a geographer, and so he was involved in some of the geographic aspects of this project. Our advisors for our particular cohort of the IGERT program were my PhD advisor, Maya Marcheva, and Craig Osenberg. And Dave Smith was also involved in many aspects of this work. As far as support, so Miguel is supported by NSF and the Puerto Rico Science, Technology, and Research Trust, and we definitely want to thank Nimbus a lot for helping to facilitate in-face collaborations on this project. Thank you.
Comments or questions? Lou always has one, so we can let somebody else go first. Shelby, but okay.
So what's the timescale that you're using in this model?
For the models, we're always looking at equilibrium dynamics here.
Okay.
So infinite, if
Okay.
that's what you mean. Or
But like over days, or like what's your time
well,
step?
the parameters are in days.
Okay.
Yeah.
But there's no inclusion of seasonality or anything like that?
No.
Okay.
No. Yeah.
Thank you.
Any other comments? Besides Lou? Oh, yeah. Oh, no, there's somebody. Wait a minute. Sorry, Eric, I'll get you in a second.
I'm curious about the airport as a spatial thing, because it doesn't seem like something that is something you visit on a daily basis or that necessarily defines a community. So how much did it contribute and to your explanation, and why did you pick it?
Yeah, so we picked it because we thought it might be associated with, the wealthier you are, the more likely you are to travel. So that was one sort of hypothesis that we had, but it turned out to not contribute as much. In fact, it contributed the least of these four. Mm-hmm.
Yeah. Eric, go ahead.
You were talking about making inferences where there were not cell phone towers. Have you done any removal of towers?
Oh,
from
we
your
haven't,
data?
but that would be interesting to look at, yeah. And that would be a good way to try and test whether that hypothesis would actually work.
I guess she gave in. So while we got this figure up, I often think that you learn a lot about data from the outliers. There's two obvious outliers there.
Yes.
Can you say anything about them?
I think it's because of the fact that when you're doing I think it's more a consequence of the modularity optimization. You can look at the actual sort of the bumpiness, I guess, of the modularity score for all these different partitions. And so I suspect that what happened here is that it just happened to be that those scores, the partitions just happened to be very different. So maybe the simulated annealing maybe wandered off in a very different location of that space compared with all of the other partitions.
But you redid the simulated annealing 100 times for each one of these partitions?
No, so we did the simulated annealing 100. So each of these is from one of the.
Oh, it's from one. Each one of those is from a single
optimization
routine.
Right.
Ah,
Yeah.
Okay. So they're not averages.
Right. So for some reason in this case, I think it wandered off and found a totally different looking partition than the others, and so so it may be that although what I would expect is that this partition, and it would be interesting to look more closely at that, that these partitions look quite different maybe from the ones over here.
And then with regard to the predictor variables, presumably there's a whole set of these things that you could have gotten data on.
Yes.
So, and I'm always worried about, well, you bias the results by choosing these over others. So did you do any kind of model selection associated with that set of possible predictor variables?
So I can't remember the things that we tried, but I think that we were looking at more at first, and then we decided to stick with these 4. But I think that part of the thing was that there was going to be a lot of correlation between different variables. So for example, for wealth index, there were other things like motorcycle ownership that would be highly correlated with wealth index. So we didn't think it made sense to have both of these quantities in there. The sugar cane, I think part of why we started looking at that is because Haitian migrant workers will often go to the Dominican Republic to work on the sugarcane over there. So I think that was one reason why we thought about this particular one.
Okay. And the data set is now about 10 years old, so you could presumably obtain, assuming the company is still around, that has the data to redo this.
Right. So actually, so Nick Ruchtenunshai, who I'm still working with, he works on cell phone data a lot, and so he has access to different data sets that, for example, one in Namibia. I believe that they might have one in Hispaniola as well. So that's something that he's continuing to work on, and he can see it a lot more refined than what we got to see here. So here everything is aggregated to the tower level for
obvious reasons.
For obvious reasons, yes. But so he's doing some interesting work on closer to the raw kind of data.
But I would have assumed that in Hispaniola that this, over 10 years, this situation has changed drastically.
Yeah.
And it'd be interesting to see if indeed the same methods lead to somewhat similar kinds of aggregation things. So even if it was just 1 or 2 other years of data, that's similarly at cell tower level.
Yeah, no, that's a very good point. That would be really interesting to look at.
I don't know, maybe there's a whole history of people doing these kinds of analysis over time, but it's a mixture of, I'll call it cultural change potentially, as well as potentially migration and other.
Right.
Cool.
Yeah, that would be interesting, yes.
Sorry.
Anybody else?
Oh, Charlotte.
Thanks, Eric. Olivia, sorry if I missed this, and I love this talk. It was so cool to see super hardcore theory combined with really modern machine learning methods applied to this, increasingly sexy area of geography and development, the use of cell phone metadata. Even though sometimes I'm sure it creeps out the public. They're like, what? They know my logs? What? It's been really powerful to see how the data has been applied to answer outstanding questions in development. And this at least was very novel for me because I hadn't seen research that integrated both mathematical theory with data-driven insights. So I apologize if I missed this. I was trying to follow along and then I kind of fell back behind the slides. But do you see a way in which this cell phone data will
speak to the types of theoretical modeling that you've done? It looks like you kind of
Sure.
have two different scales of migration that you've considered in very discrete projects.
Yes.
And I'm wondering if the cell phone data will drive your team to reconsider how those migration terms are specified or how the problem is even set up in the first place.
Yes, yeah, so yeah, thank you for that. That was a great question. Yes, so the cell phone data is, it's going to be more applicable to the short-term movement model. So that's something that Nick has worked on, is actually coming up with the parameter values for proportion of time spent in other, in different places based on that kind of call volume data. So it would be less, perhaps less useful for something like the migration, long-term migration. But what we have seen in our more recent work is that, when we incorporated the heterogeneity in movement, that didn't seem to be, with that short-term movement, it didn't seem to be as important as things like heterogeneity in spatial transmission and individual risk. And I think that's something that's bearing out of their data that they're looking at now as well. So it's good to have it to parameterize that movement, but in terms of the heterogeneity, it matters, but it's not to the same extent as the spatial transmission heterogeneity and individual risk.
So when you were doing the comparison of the prevalences with the, prevalence with the homogeneous and heterogeneous, and they were all, you had this diagonal line, right, and all the blue dots.
Right.
And then, but then when you got the next one, you said you got, then you go to the next slide. They all seem to stay below the diagonal.
Yes.
Yeah, which I wasn't sure. I didn't know that. I wasn't sure if that would happen, but they do all stay below. And then, of course, you said there. Of course, it starts to be a little bit of, non-monotone properties. But it's interesting. Were you expecting them all to stay below? I'm not 100% sure. I was expecting it.
Yeah. So, given what we had done before with the migration, we were not expecting them to all be below. We were not. Yeah. So we're still trying to think about this and understand why we're seeing these various patterns. But yeah, it wasn't something that we weren't expecting to get the totally opposite conclusion as with migration. Yeah.
All right. Any other comments or questions? Anybody? Okay. Thank you very much.
Thank you.